So after we have learned the characteristics of Trichomonas vaginalis, which is the only pathogenic trichomonad, let us know the difference between the three Trichomonas species. So the three species are Trichomonas vaginalis, Trichomonas tenax, and Trichomonas hominis. So let us differentiate the three of them with regards to their habitat, size, nucleus, undulating membrane, inclusion bodies, and the specimen used. So first, the habitat. So for Trichomonas vaginalis, it infects the genitalia of the human host. For the Trichomonas tenax, its habitat is the oral cavity. While for the Trichomonas hominis, its habitat is the intestine. So, you can really see the difference between the three species with regards to their habitat. So, malayo sila sa isa't isa. So, tenax, oral cavity, hominis for the intestines, and Trichomonas vaginalis for the genitalia. Next is the size. So the smallest of all the trichomonads is Trichomonas tenax. So it is the smallest. Intermediate, so the medium size trichomonad is the Trichomonas hominis, while the largest is Trichomonas vaginalis. So next is the nucleus. So Trichomonas tenax is the only trichomonad with a round nucleus, while Trichomonas hominis and Trichomonas vaginalis, both of them have ovoid nuclei. So Trichomonas tenax, round nucleus, Trichomonas vaginalis and Trichomonas hominis, ovoid nuclei. Okay? So next is inclusion body. So for the inclusion bodies, Trichomonas tenax and Trichomonas hominis do not have inclusion bodies. So none. Diba, they are the commensal trichomonads. So they do not have inclusion bodies. On the other hand, Trichomonas vaginalis have siderophil granules. Okay, so Trichomonas vaginalis lang ang may inclusion bodies, which are called siderophil granules. So for the specimen use, you can actually get an idea of what specimen is used to diagnose these trichomonads by looking at their habitat. Kasi diba, for example, let's start with Trichomonas tenax. So Trichomonas tenax infects the oral cavity of the human host, diba? So its specimen should be from the oral cavity. So its specimen used is oral scrapings. Okay? So diba, you get the point? Oral cavity siya. So you will use oral scrapings as your specimen for the diagnosis. For Trichomonas hominis, it is found in the intestines, diba? Intestines. So what sample will you use? You will use a stool sample. Okay, diba gets? For Trichomonas vaginalis, it is found in the genitalia. So what are the specimens that you will use? So you can use the urine specimen. You can use, for the females, a vaginal swab, vaginal discharge, or a urethral discharge. Okay? So vaginal swab, vaginal discharge, and urethral discharge for the females. For the males, you can use prostatic secretions. Kasi diba, it affects the prostate of the males. So you can use prostatic secretions. So diba, it is very very helpful to know kung asan sila nag-i-infect for us to know the other characteristics of the trichomonads. So, next is undulating membrane. So, pwede niyong lagyan dyan sa baba ng another box or another space for you to put. So, undulating membrane for Trichomonas vaginalis is less than 1/2 of the costa. For Trichomonas tenax, it is 2/3 of the costa. For Trichomonas hominis, as long as the costa. So again, for the differences between trichomonads, habitat, Trichomonas vaginalis, genitalia, Trichomonas tenax, oral cavity, Trichomonas hominis, intestines. So, memorize nyo talaga yan, guys. So for the size, the smallest is Trichomonas tenax, intermediate is Trichomonas hominis, and the largest is Trichomonas vaginalis. For the nucleus, only Trichomonas tenax has a round nucleus and the others have ovoid nuclei. For inclusion bodies, only Trichomonas vaginalis has an inclusion body called siderophil granules. For the specimens used for Trichomonas vaginalis are urine for both of male and female. For females, vaginal swab, vaginal discharge, and urethral discharge. And for males, prostatic secretions. Specimen used for Trichomonas tenax is oral scrapings. For Trichomonas hominis, stool. And for undulating membrane, Trichomonas vaginalis, less than 1/2 of the costa, Trichomonas tenax, 2/3 of the costa, and Trichomonas hominis, as long as the costa. So at the bottom side, you can see comparisons between the trichomonads. So, di ba, Trichomonas tenax is the smallest, then Trichomonas vaginalis is the largest. Okay? So, we are done with the trichomonads. So, now let us have our Q&A portion for this part. So I hope you will get a 10 out of 10 with this Q&A portion. Okay? So number 1. This is a gold standard for T. vaginalis diagnosis. A. Culture. B. Serology. C. Wet mount. D. Gensa. Answer is letter A. Culture. Diba? What culture medium will you use? Modified diamond medium, diba? So wet mount is the most inexpensive and easiest way. Okay? So I hope you got it correct. Number 2, this parasite presents with a characteristic flask-shaped ulceration. Flask-shaped. A. Trichomonas vaginalis. B. Chylomastix mesnili. C. Balantidium coli. Or D. Entamoeba histolytica. So the answer is letter D. Entamoeba histolytica. Kasi diba, Chylomastix mesnili is a commensal. So it will not form an ulceration. While B. coli and E. histolytica, both of them will present with ulceration, diba? But Balantidium coli will have a wine glass ulceration. E. histolytica, on the other hand, will have, like the question, flask-shaped ulceration. So the answer is letter D, Entamoeba histolytica. So, okay. Number 3. This parasite is a causative agent for PAM. So what is the meaning of PAM? Primary, amoebic, meningoencephalitis. So letter A, Acanthamoeba species. B, Naegleria fowleri. C, Dientamoeba fragilis. Or letter D, Entamoeba coli. So the answer is letter B, Naegleria fowleri. Because Acanthamoeba species causes GAE or granulomatous amoebic encephalitis. Dientamoeba fragilis will cause colicky abdominal pain, diba? And Entamoeba coli is a commensal, so it does not cause any clinical manifestations in the human host. Okay, so did you get it right? So number 4, which trichomonad uses oral scrapings for diagnosis? A. Trichomonas vaginalis. B. Trichomonas tenax. C. Trichomonas hominis. Or D. Trichuris tricura. So the answer is Trichomonas tenax, diba? Because Trichomonas vaginalis will use urine, vaginal swab, vaginal discharge, urethral discharge, and prostatic discharge as a specimen of choice, while Trichomonas hominis and Trichuris tricura will use stool samples for its diagnosis. Okay, so the answer is letter B, Trichomonas tenax. Next, what inclusions are found in Entamoeba histolytica trophozoites? A. Rounded chromatoidal bars. B. Splintered chromatoidal bars. C. RBCs. Or D. Platelets. So you can answer. The answer is letter C. RBCs. So diba, rounded chromatoidal bars are found in Entamoeba histolytica cysts. Diba? Kasi kung chromatoidal bars, you can find it sa cyst form. Splintered chromatoidal bars are found in Entamoeba coli cysts. So splintered chromatoidal bars are witch broom chromatoidal bars. Yung rounded chromatoidal bars, letter A, also called as sausage-shaped chromatoidal bars. So answer is letter C, RBCs. Diba? It is the only amoeba which has RBC inclusions in its trophozoites. So yung platelets, nandyan lang yan siya. So, okay, letter C, RBCs. Number 6, what is the largest intestinal protozoan? A. B. coli, B. E. coli, C. E. histolytica, or D. Dientamoeba fragilis. So the answer is letter A. B. coli. So it is the only ciliate known pathogenic to humans, and it is also the largest intestinal protozoan. So the answer is letter A. B. coli. Okay, so next, this is the reproductive nucleus of Balantidium coli. A. Macronucleus or B. Micronucleus. So the answer is letter B. Micronucleus. Kasi diba, letter A, macronucleus is a kidney-shaped nucleus and it is the vegetative nucleus of Balantidium coli. While the micronucleus, the answer is the reproductive nucleus, and it is shaped, characterized as dot-shaped nucleus. Okay, so the answer is letter B. Number 8, the trophozoite of this flagellate is characterized as shepherd's crook in appearance. A. Giardia duodenalis. B. Chylomastix mesnili. C. Trichomonas vaginalis. or D. Trichomonas tenax? So the answer is letter B. Chylomastix mesnili. Diba, Giardia duodenalis, trophozoite is old man with eyeglasses, tennis racket without handle, or smiling shape trophozoite. While for Trichomonas vaginalis and Trichomonas tenax, they are pear-shaped trophozoite with jerky motility. While for Chylomastix mesnili, Shepherd's crook trophozoite, then ang cyst niya is lemon-shaped or nipple-shaped cyst. Okay, gets? So answer is letter B, Chylomastix mesnili. Next, number 9. These are protozoans with pseudopodia. So these are classified under A. Sarcodina or B. Mastigophora. Answer is letter A. Sarcodina. Kasi diba Sarcodina, amoeba, pseudopodia. While Mastigophora, flagellates. So with flagella. Example, Trichomonas, Giardia, they are classified under Mastigophora. But for pseudopodia, amoeba, classified under Sarcodina. Okay, so answer is letter A, Sarcodina. So number 10, a formed stool sample is passed to the lab. Which of the following will you most likely see during microscopy? A. E. histolytica troughs. B. E. histolytica cysts. C. T. vaginalis troughs. Or letter D. T. hominis cysts. Answer is letter B. E. histolytica cysts. Kasi diba, formed stool sample. So most likely, you will see cystic forms of the protozoans. So, hindi siya letter A because trough siya, diba? And letter C, T. vaginalis trough. So trough and T. vaginalis, it does not use stool samples, diba? As diagnostic specimens. While letter D, Trichomonas hominis cysts. Bakit hindi ito, diba? Cyst siya. And Trichomonas hominis uses stool samples. But mind you, diba, Trichomonas hominis does not have a cystic stage. So, trophozoite lang siya. So, the answer is E. histolytica cysts. Okay? So, I hope you got 10 over 10 with that Q&A portion. So, di ba, easy lang siya once you take a grasp of the very important concepts. Okay? So now, we are done with the trichomonads. Let us go into the blood and tissue flagellates. So blood and tissue flagellates. Hemoflagellates includes what two genera. So the two genera are Leishmania, and Trypanosoma. Diba? Trypanosoma. So, Leishmania and Trypanosoma. So, these can infect blood, lymph nodes, muscles, and the reticuloendothelial system. So that's why they are called blood and tissue flagellates. Okay? So these blood and tissue flagellates, so both Leishmania and Trypanosoma, have their stages of development. So first is the amastigote, or what you call the Leishmania or Leishmaniad form. Leishmaniad form. So this is the ovoid form and devoid of flagellum. So you can see it in the picture, ovoid and devoid of flagellum. Next is promastigote or leptomonad form. Promastigote or leptomonad form. It is elongate. Its kinetoplast is anterior to the nucleus, so as you can see sa picture, diba? And its flagellum is at the anterior end of the body. So next, epimastigote, or what you call the crephidial form. So crephidial form. So it is elongated with a juxtanuclear kinetoplast with a short flagellum. So juxtanuclear kinetoplast, malapit sa nucleus ang kanyang kinetoplast, diba? As you can see sa picture. Then the last stage of development is the trypomastigote, or what you call the trypanosomad form. So its kinetoplast is post-nuclear, so sa baba siya ng nucleus, and it has a long flagellum almost the same size as its body. Okay, so those are the stages of development of the blood and tissue flagellates. So I hope you will really remember them. Not just memorize, but familiarize yung mga mukha nila so you can imagine them when you take the board exam. Okay? So, we will go first to the trypanosomes. So, di ba, trypanosoma and Leishmania. So, first, we will go to the trypanosomes or the trypanosoma species. So, there are two types of trypanosomes, stercorarian and salivarian. So stercorarian trypanosomes are found, kaya siya stercorarian kasi it is found in the stool or in the parang ini-inject niya yung stool, or in fact ang stool ng vector is infected with the trypanosomes. But with salivarian trypanosomes, it is found in the saliva mismo of the vector. So stercorarian, it is due to the contamination of the stool of the vector. So the stercorarian trypanosome is Trypanosoma cruzi, while the salivarian trypanosomes are Trypanosoma brucei gambiense and Trypanosoma brucei rhodesiense. So as you can see, lahat ng mga brucei, they are salivarian trypanosomes. But Trypanosoma cruzi is a stercorarian trypanosome. Sterco, diba? Stool, sterco. Okay? So first, let us go to Trypanosoma cruzi. Okay? So, Trypanosoma cruzi is the agent of, na-discuss na natin to kanina, it is the agent of Chagas disease and American trypanosomiasis. So, Chagas disease and American trypanosomiasis. Okay? So, it is characteristic okay? So, the characteristic shape of trypanosomiasis